Okay? So I promised we'd start with the replication fork, and again, I think this should be review for most of you. And so this is the site at which DNA replication occurs in all chromosomal DNA replication, with very few exceptions. The exceptions would be some viruses and some transposases, things like that, that have very unusual means of replication. But for the most part, any cellular replication is going to occur at a structure like this. Okay, so I'm just going to introduce some basic terms. The first is the leading strand DNA synthesis. And actually, I'm going to introduce you to these in terms of the enzymes involved. Okay, so we're going to talk about all the enzymes that act at a replication fork. And the first one we're going to start with is by far the easiest one to understand. It's the leading strand DNA polymerase. It moves in the same direction as the overall replication direction. Okay? So it's easy to understand because it basically needs one primer at the beginning of the process. And assuming you have adequate processivity, that primer will be sufficient to completely replicate until you either hit the end of the DNA or reach another replication fork coming from the opposite direction. Okay? So this is the most straightforward. So I think the key thing is this moves toward the unreplicated DNA. Okay, so the second enzyme that acts at the fork is another DNA polymerase. Okay, and it's going to turn out that in some cases these are the same molecule, in other cases they're different. But we're going to call this the lagging strand DNA polymerase. And it moves its direction of synthesis. Okay, when it extends the 3' end of a primer, it's going to be moving away from the unreplicated double-stranded DNA. Okay? As opposed to here, when you extend the 3' end of this primer here, it's going to move this direction. And because of the anti-parallel nature of DNA, this has to be the case. You cannot have both of them moving towards the unreplicated DNA, however much that might seem nice. So this creates our first problem, which is that in order for the lagging strand to be synthesized, you have to do it piecemeal. That is, if you're going to have this polymerase moving continuously, this one's going to have to make little chunks, which are referred to as Okazaki fragments or lagging strand DNA fragments, a little bit at a time. Okay, so this one, for example, would stop here when it reached this primer because there's nothing left for it to replicate. There's no more primer template junction, right? No single-stranded DNA. And it would have to fall back off. There'd be a new primer here and start over again. And we're going to come back and talk in much more detail how the cell deals with that problem. But that's a critical issue that the cell has to deal with. Okay, so the next enzyme that's required for both the leading and the lagging strand synthesis is primase. Okay, and primase is actually not a DNA polymerase, but an RNA polymerase. So it's a specialized RNA polymerase. And it synthesizes 6 to 8 base pair RNA primers at replication forks. Okay? And the reason it synthesizes them at replication forks, well, actually, I'll come to that in a second. So the reason you need primase is the following. DNA polymerase cannot start a new DNA strand from 2 nucleotides. Okay? It must have a primer. Okay? It can't take two nucleotides and put them together and then add a third, a fourth, a fifth, a sixth. It has to have a primer. That's why I said yesterday or Tuesday that we needed a primer template junction. Okay? Now it turns out that that primer can be either RNA or DNA. And the other important thing to know is, unlike DNA polymerases, RNA polymerases can take two ribonucleotides, put them together, and start a chain. Okay? So they can take two ribonucleotides, put two together, and then add a third to that, and a fourth, a fifth, a sixth. And in this case, on average, it'll make 6 to 8 of these. Given that it typically makes 6 to 8 bases of RNA primer, what kind of processivity does this enzyme have? Really low, right? So you could think about, how would I assay the processivity of primase? It's an interesting question that you could maybe run into on an exam or a question at some point. But you know, we're not going to teach you how to do everything here, but we want you to learn to generalize the assays that we've taught you already in other settings. Okay, so that's an example of something like that. Okay, so the other interesting aspect of this is this first thing that I said, which is that it acts at replication forks. And it turns out the reason primase acts at replication forks is it is activated by interacting with another replication fork enzyme and that's called DNA helicase. And I should emphasize that this is true in bacteria. We'll talk about eukaryotic cells later. They actually carry their primase firmly coupled to one of the lagging strand DNA polymerases. Okay? But in either case, it's restricted to acting at the replication fork because you don't want to start priming DNA synthesis willy-nilly all over the genome, because you would get all kinds of bubble-shaped DNAs that would not be good to have. Okay, so there's DNA primase on this image. And now I want to turn to the next enzyme, which we'll spend significantly more time on, DNA helicase.